Hello everyone. In today's class, we will be learning the basic requirements and the techniques of plant tissue culture. Let us see what are the content we are going to deal with. We will start with the concepts of PTC, that is plant tissue culture, and the instruments that are needed for the plant tissue culture. Then the sterilization of the glassware, media and the equipments for the need of plant tissue culture. Lastly, we will deal with the major techniques involved in plant tissue culture. Now, we will learn the concepts of plant tissue culture. We all know that plant tissue culture is the heart of biotechnology. Plant tissue culture is the technique that uses an artificial media to grow and maintain the plant cells and tissues under controlled environmental conditions. Plant tissue cultures are now becoming a very common technique that has been adopted for propagation of useful plant species by in vitro conditions. Plant tissue cultures are also grown in manipulating the gene to obtain the plant secondary metabolites, that is, their active metabolites can be obtained using plant tissue culture technique. To perform plant tissue culture experiment, a tissue must be obtained from a plant. The tissue obtained from a plant to be cultured is called as an explant. Explant regenerate root and shoot system, then consecutively the whole fertile plant can be obtained under certain cultural conditions. Several parts of the plant like leaf, shoot tip, root tip, growth buds or apical meristems can be used to initiate the plant tissue culture. However, shoot tip and auxiliary buds are commonly used in micropropagation techniques. Plant cells have a unique property called totipotency. Totipotency is the ability of a single cell to divide and produce all the differentiated cells in an organism. That is, totipotency is an important concept of plant micropropagation. If any part of a cell is excited and placed in a suitable cultured condition with carbon source, growth regulators and vitamins, the cell will undergo mitosis. Later, depending on the concentration of the auxins and cytokine present in the culture medium, will encourage the root and shoot growth respectively. This property makes it possible for us to use several parts of the plant as a source of an explant. Let us start with the laboratory requirements, among which the first is the laminar airflow bench. Since the growth of the plant cells and tissues are propagated in strictly controlled conditions, the process of plant tissue culture should only be operated under absolutely sterile conditions. For this, laminar airflow bench is very necessary. This laminar airflow bench is fitted with HEPA filter, that is high efficiency particle air filter, which efficiently block all type of microbial organisms. Air is sucked into this filter and flow over the working area in laminar airflow. This will ensure that the work area is free of microbial contaminants. This laminar airflow is also fitted with germicidal UV lamp, which is also always switched 10 to 15 minutes before every session of culturing. The next requirement of plant tissue culture are autoclaves. All the glasswares and the solutions used for plant tissue culture should be sterilized before use. The solution and the culture vessels used for plant tissue cultures are generally sterilized using an autoclave. But the temperature sensitive component in solutions like growth promoting hormones should be filter sterilized and they should not be autoclaved. The next item is culture vessel. The container in which the explants are cultured are called culture vessels. They are test tubes, bottles, wide mouth troughs. All the vessels are fitted with lids that fits tightly. Additionally, they may also be fixed with membrane filters through which air exchange can occur while maintaining sterility. All the vessels are autoclavable and can be individually sterilized before use. Normally, the growth medium is prepared and aliquoted in the culture vessels in the required volume. Then the containers are closed with the lids and properly autoclaved. During autoclaving, the lids are fixed on loosely and they are tightened after the autoclaves cool down. Now let's see the dissection instruments needed to handle the plant explant. The explant used for plant tissue culture are derived from the plant themselves. For this, the plant source has to be collected and brought to the laboratory. In the laboratory, they has to be surface sterilized and dissected to prepare the explant to be cultured. To cut the explant, the scissors and scalpels are used. To prepare the explant to be cultured, the bent forceps and sharp forceps are used. Now let's see the role of incubation racks in plant tissue culture techniques. In plant tissue culture, the cultures are usually incubated for long period, extending over several weeks. Additionally, the growing cultures are autotrophic, which means they start carrying out photosynthesis, with the exception of some callus cultures and adventitious root cultures. Thus, there is a dire requirement for the light for the photosynthesis. Efficient photosynthesis can be mediated by alternating light and dark period, as in occurring in the nature environment. This is facilitated by fitting fluorescent lamp in the incubation rack used for PTC. The lights are fixed at distance of 10 to 20 centimeters from the culture vessels for maximizing the incidence of light on the culture. Timers are also fitted to control the light and dark period duration. This is called light cycle or a photocycle. Most commonly, 16 hours photoperiodism and 8 hours dark period is used. This photocycle can also be optimized for different cultures. In cultures that are established well for mass production, special equipments called bioreactors can also be used. After the plantlets are developed in culture, including induction of shoot and root using shoot-root inducing medium condition, they need to be shifted to field condition. But being nurtured and pampered in strictly controlled laboratory conditions, these plantlets will be more vulnerable to environmental shock when moved out of the laboratory conditions. These plantlets are transferred to sterile soil in plant tissue culture laboratory. They are acclimatized for few days and then transferred to the chamber. The chamber is a real environment. Hardening chamber requires two major infrastructure requirements, namely greenhouse and mixed chamber. In present days, commercial manufacturers are combining the two to provide greenhouse mixed chamber. Usually, the greenhouse mixed chamber is constructed using glass and sprinkling system with high humidity monitored conditions. More commonly, in our country, hardening chambers are constructed with using metal structures that are fitted with green netted sheets. They are used in acclimatizing the plantlets. Later, these plantlets are shifted to the similar structures with a transparent plastic sheets instead of a glass. After hardening, the plants are ready to take to the field condition, where they can continue to grow on their own without any medium support. Now let's see the most important requirement for the plant tissue culture, that is the media. The plant tissue culture media should generally contain some or all the following components. The plant tissue culture should contain the inorganic salt, that is the macro element. It is also called as major salts, and micro elements, it is also called as minor salt. It should also contain a carbon or an energy source. Usually sucrose is considered, followed by vitamin, amino acid, growth regulators. Growth regulators are also called as plant hormones or their analog, and gelling agent like agar, agarose, etc. The most commonly used plant tissue culture medium is MS medium, otherwise called as Murashi Scoops medium. Besides carbon, hydrogen and oxygen, the essential element in plant cells or plant tissue culture media includes the inorganic element that are required in higher concentration in the medium. They are referred as macro element in the medium. They are added as the salt of the elements. Besides they are added at a higher concentration, they are called as major salts. This contains 6 elements, namely nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. As for the minor salts, the inorganic element that are required in lower concentration are called minor salt, and the stock of the solution of the salt is called as minor salt solution. The minor salts includes iron, manganese, zinc, boron, copper, molybdenum. Though they are used in low quantities, these elements are absolutely essential for the plant growth and therefore vital components for the plant growth medium. Now let's see the carbon and the energy source which are essential for the plant cell culture media. Sucrose is the most commonly used carbon source in the plant tissue culture medium, though other sugars like lactose, maltose, galactose, raffinose, trehalose and cellulobiose have also been tried. The best way is to use sucrose in the medium is to add the sugar in the prepared mixture of the medium and then autoclave it. This causes the hydrolysis of the sucrose to its component sugars of glucose and fructose, which seems to be very ideal source for that particular medium. The use of the individual sugars, that is glucose and the fructose, ironically does not work satisfactorily as that of sucrose. Similarly, the filter sterilization of the sucrose solution before addition into the medium as an alternating for autoclave also does not work satisfactorily. In order to induce the plant cells or explant to start multiplying and growing in cultures, a variety of growth regulators are used. Normally, they constitute the plant hormones or phytohormones. They are auxin, cytokinin, gibberellin and abscisic acid. Now, let's see the properties and function of auxin in the plant cell division. Auxin can induce cell division. They can cause shoot formation and shoot elongation. They help in callus formation as well as root induction. Depending on the derivatives and the concentration, the function properties varies. The most commonly used auxin is indole acetic acid, called as IAA, or its naphthenic derivative NAA, for cell division and shoot elongation. To induce callus formation, another auxin called 2,4-D, called as 2,4-dichlorophenoxyacetic acid, is commonly used. Now let's see another growth regulator, cytokinin, which helps in shoot differentiation. Cytokines are used for cell division as well as in somatic embryogenesis process. They can also increase the metabolic activity of the cells in the culture. The most commonly used cytokinin are BAP, that is benzylaminopurin, and kinetin. Usually, a combination of auxin and cytokinin is used in plant tissue culture to induce shoot, root development. Now let's see how the ratio of each of this growth regulator component is used to decide which process has to be induced in the culture. The ratio of the growth regulators vary among morphogenesis, embryogenesis, callus initiation, root initiation and shoot initiation. Generally, the initiation and maintenance of the callus culture requires both auxin and cytokinin, while only auxin is essential for the root culture and only the cytokinin for the shoot culture. The concentration of the plant hormones used also depends on the type of the explant used. In addition to the auxins and cytokinin, gibberellin and abscisic acids are also used for specific purposes in plant tissue culture. The most commonly used gibberellin is gibberellic acid 3. Apart from the above discussed component, the media used for plant tissue culture is also supplemented with other components like vitamins, amino acids and some organic acid. The mature plant cells are capable of synthesizing all or the most of these components, but the immature cells are coaxed for the growth in culture needs to be supplemented with these components. The major vitamins added to the media are thiamine, riboflavin, niacin, pyridoxine, folic acid, pantothenic acid, biotin, ascorbic acid, tocopherol, myoinositol and para-aminobenzoic acid. Nitrogen source in the media is sometimes provided as amino acid form. Though ammonium salts are most commonly used, it is sometimes observed that nitrogen is more readily available for amino acid than from the ammonium salt. Again, this differs with the type of cultures and the plant species involved. The commonly used amino acids are casein hydroxylase, L-glutamide, L-asparagine, adenine and ammonium salts. Organic acid like pyruvate and TCA cycle intermediates are also added to the PTC medium as a supplement. Apart from this, the complex mixtures like coconut milk, casein hydroxylate, yeast extract, tomato juice are also sometimes added to the medium. It will promote the cell division and the growth as they contain several growth factors. However, these mixtures are not very preferred by many because of the variation in the quality and the quantity. Sometimes to promote growth, activated charcoal is added to the medium. This promotes the growth by adsorbing some inhibitory components like phenols. These phenols are released by the growing cells themselves. In addition to this, all time some antibiotics may be added to the medium to control the growth of microorganisms. Apart from the nutrition and growth promoting components, we also need to add innate supporting materials or gelling agent in the medium. The most commonly used gelling agent is agar. Gelatin, phytogel, agarose and other such gelling agents can also be used, but agar is more satisfactory. So far we have seen the types of media and the supplements needed for the plant tissue culture. Now we will know the sterilizing agent for the plant explant. The explant used for the plant tissue culture is usually collected from the field or from the wild areas where the plants are exposed to the environmental stress, air, waterborne microorganisms, airborne microorganisms and many plant pathogens. So this explant has to be immediately immersed into the sterilizing agent and they have to be thoroughly washed out. The most commonly used surface steriling agents are hypochlorite, mercury chloride, 70% of ethanol, silver nitrate and hydrogen peroxide. It is also highly desirable to use a detergent before the surface sterilized to increase the wettability. The choice of the sterilization agent and its concentration depends upon the type of explant we use. Even though many different types of media have been used in plant tissue culture, the most commonly used media is MS media, which was devised by Murashi and Scoop. It contains specific volumes of stock solutions of macroelements, that is major salt solution, and it also contains microelements, minor salt solution. It also contains vitamins, amino acids and sucrose as a carbon source. The required phytohormones are also added. The pH of the medium is adjusted to 5.6 to 5.8. After adding all the components, these components are mixed well. A solidifying agent, that is agar, is added and the mixture is heated to about 60 degrees centigrade to dissolve the agar. While the mixture is still warm, it is aliquoted into the culture vessels at the required volume. Approximately 20 ml medium is added to each culture tubes and 25 to 40 ml medium is added to each culture vessels. Based on the size of the flask, the medium has to be added. Immediately after adding the media to the tubes are cotton plugged and the flask are covered loosely with the screw cap lids. They are autoclaved. After autoclaving, the tubes or the flask are allowed to cool under sterile conditions. Once cooled to the room temperature, the lids are tightened and the medium can now be used for initiating a new culture or as well as for transferring the cultures for the subculturing processes. Let's learn the methods of sterilization of explant. Irrespective of the type of explant used, if they are collected from the plant grown in the field or from the wild areas, they need to be thoroughly surface sterilized. If the explant has a waxy coating, one should also use wetting agent before the sterilant to ensure the good contact of the sterilant with the surface of the explant. The commonly used wetting agents are tween 20 T-Pol solution or alcohol, that is 70% ethanol. Normally, the explants are immersed in the wetting agent for 15 to 20 minutes. Later, the duration of exposure also depends on the type and the nature of the explant. More often, the explants are immersed in detergents for 15 to 20 minutes and then transferred to 70% of alcohol for 60 seconds before being transferred to the sterile solution. Several types of surface sterilizing agents can be used with the explants. As discussed earlier, sometimes combination of surface sterilizing agents like mercury chloride and sodium hypochlorite can be used. The duration of the exposure to the sterilizing agent is also depending on the explant type. During the sterilization process, the bottle containing the explant and the sterilant is whirled frequently to enhance the effectiveness. After this process, it is very vital to remove all the traces of sterilizing agent by extensive washes of several changing of distilled water. Now the explant is ready for the culture. Now let's see the method of inoculation of the explant into the media. The prepared explants are carefully and gently lifted using sterile blunt forceps and transferred onto the growth medium prepared earlier. The explants are pushed partially into the medium instead of just overlapping. Complete immersion of the explant is avoided. This ensures that this explant is in good contact with the medium and also with the exposure of air in the container for the gases exchange during their growth period. After inoculating the explant into the media, the explant need to be exposed to an adequate light for optimum growth. This is ensured by providing a 16 hours light and 8 hours dark photocycle using a timer that was controlled by fluorescent lamp fitted onto the incubation rack. During the photo period, light of 4 to 10 into 10 to the power of 3 lux light intensity is provided. For seed culture and callus culture, complete darkness is preferable, which can be achieved by placing the culture vessel in a dark box or by covering the bottle with a dark paper before placing in the incubation racks. The incubation racks are usually placed in a room that is maintained in 25 to 28 degrees centigrade. The plant culture usually takes several weeks to develop. During this period, it is very essential to monitor them carefully for any contaminations or changes. If a culture is appearing unhealthy, it should be removed immediately to ensure that the adjacent cultures are not getting affected. Now let's see how propagation and subcultures are done. A newly initiated cultures usually grown first by shooting. This means that the shoot system propagates first. Each explants can give rise to several shoots. As this takes place 2 to 7 weeks time, medium depletion may occur. It is therefore important to make a subculture now to grow a explant into a fresh media. Usually from a single explant, multiple shoots develop. They can be prized now and carefully inoculated into fresh media for further growth. This process is called subculturing. One single explant can give rise to hundreds and thousands of plants by subculturing process. Similarly, the callus tissue also need to be subcultured for the further maintenance. Now let's see how to subculture the callus to the fresh media. For this, the callus is cut into pieces under sterile condition using sterile scalpel and minced gently into small pieces. Each of these can propagate into a large masses of callus. From this, callus tissues and the whole plantlet can develop, and now the mass propagation can be induced later by differentiation and organogenesis process by adding certain organic substances and natural complexes. After several subculturing of the callus into the media, the cells have to be differentiated and the organs has to develop. For that, the growth regulators has to be added in different concentration. Higher concentration of cytokinin induces the shoot formation from callus, while higher concentration of auxin is needed for root induction. Medium level of both cytokinin and auxin induces both shoot and root formation. Medium level of cytokinin with low level of auxin induces dedifferentiation and results in callus formation again. Gibberellins are used for shoot elongation and leaf generation. This picture shows how the inorganic substances, organic substances, vitamins, amino acids, growth regulators and some other innate supporting materials helps in the development of explant to form a callus. From the callus, they are differentiated into root and shoot, and at last the organogenesis takes place. Finally, the plantlets had developed. After differentiation and organogenesis process, plantlets are developed. We all know that the plantlets produced in the cultures are propagated in highly controlled and pampered conditions. They cannot be transferred directly to the field, where the conditions are often harsh. Therefore, these plantlets produced in the cultures need to be prepared for the field condition, and this process is called as hardening. This is preceded by a pre-treatment stage, when these plantlets are transferred into compost soil and kept in high humidity conditions in a mist chamber. Once they have acclimatized, they are transferred to the pot containing soil for a short duration. These pots are maintained in the mist chamber and later transferred to the greenhouse. Once hardened, these plants are ready to transplant into the field. So we have come to the last part of the video. So far we have dealt with the basic requirements, techniques and the medias required in plant tissue culture. Now we will be seeing the several benefits and the advantages of plant tissue culture. They include mass production of plants, improved traits in plants. They also help in the production of virus-free and the disease-free plants. They help in the development of plant tissue banks, that is by freezing the regenerated tissues or artificial seeds. They help in production of genetically identical plants by somatic embryogenesis. They help in mass production of specific secondary metabolites. They help in year-round production of plants and their products. At last, they help in the preservation of endangered plant species. In the next video, we will be learning the process of micropropagations of plant. Thank you.
Want to know more about these topics? Please follow us on Twitter, Facebook, Instagram, and don't forget to subscribe our channel.